for the treatment of the Duchenne muscular dystrophy. Dr. Aoki is Director of Molecular Therapy at the National Institute of Neuroscience and the National Center for Neurology and Psychiatry. So welcome, Dr. Aoki, and you have 20 minutes. So we look forward to hearing your presentation. Thank you.
Okay, wonderful. Thank you so much for your kind introduction, Dr. Childers. So hi everybody. I'm Yoshitsugu Aoki from the Department of Molecular Therapy at NCNP Japan. So I'd like to express my gratitude to Dr. Childers and other organizers for providing me with this wonderful opportunity to present at a past webinar. So this is a disclosure slide for my talk. So as other speakers have already presented, so DMD is the most common and severe genetic muscle disorder that primarily affects males. It is caused by a mutation in the DMD gene, which encodes the dystrophin protein, so resulting in progressive weakness and the loss of skeletal, cardiac, and pulmonary muscles. Currently, corticosteroids are used to slow the progression of the disease. So currently, there are several therapeutic approaches being investigated to cure DMD, which can be categorized into three groups: gene modulating therapies targeting DNA or RNA, new drug designs such as corticosteroids, and stem cell-based therapy. Among these approaches, the most advanced is the splice switching therapy, also known as exon skipping. So our research using DMD animal models has shown that exon skipping is feasible in dogs. This is dystrophic dogs, and mice, leading to very promising results. We have created an in vitro assay system using human fibroblast from DMD patient to test the effectiveness of exon skipping drugs. Based on these accomplishments, successful exon 53 skipping trials have been conducted in Japan and the United States. So Viltolarsen, this is our exon 53 skipping drug for the treatment of DMD, was recently approved for accelerated approval based on the observed increase in dystrophin production, so in skeletal muscles of DMD patients in Japan and the United States. So this is the data, who received 80 mg per kg of morpholino-based exon 53 skipping drug, and they received weekly injection of this drug for 24 weeks. In this study, participants with DMD were treated systemically with, as I mentioned, Viltolarsen at 40 or 80 mg per kg for 12 or 24 weeks, which led to the production of new dystrophin protein. On average, there was a 4.8% recovery in dystrophin expression, and improvement in timed function tests were also observed. So this is our drug. In the post-market analysis, more than 100 patients in Japan have undergone the treatment. At NCNP, this is our facility, 21 patients aged 2 to late 20s received treatment. No serious adverse events occurred. Although the efficacy is challenging due to the wide age range, CK levels showed a rapid decline, 50 to 60% reduction compared to baseline. Ambulant cases showed maintained 10-meter walking speed and NSAA North Star score, with some patients showing improvement. These suggest that Viltolarsen may be effective considering the natural history of DMD. However, the current oligonucleotide-based exon skipping approach for DMD has a limitation, as it is specific to certain mutations. The DMD gene is made up of 79 exons, and deletion mutations often occur within exons between 45 to 55. Viltolarsen, a current treatment is only effective. So this is Viltolarsen, which is also called NCNP01, could rescue only about 8% of DMD patients. To address this limitation, we are working on developing a drug that can skip exon 44 as a next target, which is called NCNP02, or so now we are calling it as Brogidirsen, which would potentially be applicable to around 6% of all DMD patients. Additionally, we are now developing drugs that can skip exon 45, 50, or 55 to further broaden the applicability of treatment. This is ongoing in Japan and the United States. We investigated aerosols that were 20 to 30 oligonucleotide long and targeted 2 sequences in exon 44. This is now called dual targeting oligo. So we assessed the ability to skip exon 44 in RD cells. The 24 nucleotide NCNP02, so-called Brogidirsen, demonstrated a dose-dependent increase in exon skipping effectiveness with an EC50 value of 0.83 micromolar per liter. So I didn't include the data this time. So we conducted 3 screening steps to define the sequence. And this involves using tool method, and also PMOs of different lengths, which target various regions of exon 44 of the DMD gene. During a 13-week study, homologous monkeys were given NCNP02.
Oh, okay. So this is a monkey study to check the safety of a drug. And so in this case, we used 60 mg per kg or 2000 mg per kg. It is very high dose. And so monkeys received weekly injection of this drug for 13 weeks and 8 weeks. So from the final injection, so we checked the induction level of exon 44 skipping by RT-PCR. And this data suggests, exon 44 skipping was induced dose dependently, and at 2000 mg per kg, so exon 44 skipping levels reached 17% in skeletal muscle and also around 1% in cardiac muscle. So I'd like to say this is wild type monkey. Okay. So AMED, the largest funding body in Japan, has recently granted as a project grant to develop a highly efficient exon 44 skipping drug for DMD. So in collaboration with Nippon Shinyaku, company, we decided to develop Brogidirsen, as the world's first exon 44 skipping drug. This drug induces highly efficient exon 44 skipping, and dystrophin protein expression in fibroblast cells. So this is mildly converted fibroblast from DMD patient amenable to exon 44 skipping. This could potentially slow the progression of the disease. And our drug Brogidirsen is used to artificially remove exon 45. So in the case of DMD, with a deletion of exon 45. And correcting the reading frame of amino acid, and we can expect the production of a little bit shorter but functional dystrophin. After conducting a phase 1 first-in-human trial, we proceeded with a next phase, open-label study. Brogidirsen was administered intravenously every week for 24 weeks to 6 ambulant DMD patients aged 4 to 10 years old at NCNP hospital and another hospital in Japan. The purpose of this study was to evaluate the safety, pharmacokinetics, dystrophin expression levels and motor function after the treatment. So this is Western blotting data. So using biopsied muscle samples from all the subjects. Surprisingly, we detected very high levels of dystrophin restoration. So please see those very clear band. And so at the baseline, we still see some dystrophin expression around 5%. But after the treatment for 6 months, so dystrophin increased up to 15%. So in the subject who received 80 mg per kg for 6 months. And the motor functional scores of PAL, PAL upper limb assessment and North Star ambulatory assessment indicate a maintenance or improvement trend in their motor function in the phase 2 study. So the study reviewed, this is a summary slide. And so the study revealed an increase in dystrophin protein expression on Western blot, showing an average of 10.27% of normal in cohort 1, so who received 40 mg per kg for 6 months, and 15.79% of normal in cohort 2, so who received 80 mg per kg. No discontinuation of administration occurred due to adverse events during the trials. So Nippon Shinyaku Drug Company, which is our collaborator, is preparing for the next phase of the trial based on these results in the United States and Japan. Additionally, the 6 participants are part of an extension study conducted by the company to further evaluate the efficacy and safety of continued administration. So we are investigating the amount of dystrophin needed to improve skeletal muscle function based on previous references. And it was found that Duchenne and its milder form Becker muscular dystrophy can be categorized into 4 clinically useful groups based on the correlation of clinical and biochemical data. And the data suggests that restoring dystrophin to over 10% of normal levels, as measured on Western blots, is expected to moderately improve skeletal muscle function. And so this time, we believe we achieved the level of 50% of normal dystrophin restoration in the subject, so in the cohort 2. Next, we are working on predicting the in vivo efficacy using an in vitro system, specifically mildly converted urine-derived cells. Now I call it as UDCs. So UDCs are mixed population of either uroepithelial or renal epithelial cells expressing most mesenchymal stem cells and peripheral cell markers. We have recently developed a direct reprogramming technology and created a cellular skeletal muscle model for both healthy individuals and DMD patients using mildly converted UDCs. And we have confirmed successful induction of exon 44 skipping in DMD models using mildly converted UDCs from enrolled subjects through an in vitro assay system. So the quantitative data from RT-PCR and Western blot confirmed highly efficient exon 44 skipping and dystrophin restoration in a dose-dependent manner. Also, still preliminary, our in vitro data seems consistent with efficient in vivo data observed in the phase 2 trial. And this is a final slide. So recently we have developed an e-skip finder. So this is opened, so you can use it on the internet. And this utilizes information from the literature to create a database of exon skipping antisense oligonucleotides and a predictive tool for skipping efficacy, assisting our researchers in designing effective exon skipping therapies, and this could overcome so for our too much effort and too long time to develop a new drug. So we'd like to accelerate drug development for DMD. So this slide displays the contributors to this work. So I believe that our data will be so informative for you. Thank you for your attention. And this is my mentor, so Dr. Shinichi Takeda. I succeeded his laboratory in 2019. Thank you.
Thank you, Dr. Aoki. Fantastic. Congratulations on this work and congratulations on moving this forward into the clinic.
Thank you.
Are there questions for Dr. Aoki in regards to his discussion? Yes, Dr. Townsend.
Very nice work. I really appreciate it. Looks like you're getting a little bit of cardiac expression, but I'm curious how you see this moving forward in the heart. Does exon skipping have a future for treating cardiomyopathy, or are we going to have to go with alternative therapies?
So I believe so, and I'd like to believe so. So, but as long as we use naked morpholino as a chemical, and we can inject at 80 mg per kg as a therapeutic dose, it should be very difficult to rescue dystrophin expression in cardiac muscles. And so to overcome this limitation, I'm now collaborating with Professor Matthew Wood at Oxford and try to develop a highly safe and efficient peptide conjugated PMO to rescue cardiac muscles. That's what we are doing. Thank you for your question.
Got it. Okay, thanks.
Other questions for Dr. Aoki?